Okay, now we're going to show you how to do gram staining. It's a really simple procedure. First, some of the stuff you'll need crystal violet, iodine, destaining solution, and the counterstain safranin. You need a burner, you'll need a loop, you'll need the cells that you want to actually gram stain. You'll need a slide, and notice no cover slip. You don't need a cover slip when you're doing gram staining. And then one of these trays that has a little holder on it, which is just a couple pipettes stuck together, where you can do all of your manipulations and catch all the waste in the tray. So the first thing you do is turn on your flame and take some water, and you're going to add a little drop of water to the surface of the slide. Now you can do it in a couple ways. You can get a little stream of water going like this and try to drip it on. What I tend to do is get a little on my pinky and I just drop it on the surface, because that way it gets to be a pretty small drop. You want something small, otherwise it will take 6 years to dry down. You then use sterile technique, sterilize your loop, and take the colony you want to check, and you want to take part of a colony. You don't want to take a whole bunch of stuff, and you don't need a lot of colony at all. Teeny amount is all you need. You don't need a lot of cells. Pick up your slide, and then go ahead and smear your little bacteria, the loop, into that water. And the less water you have, the better, just because it'll, again, dry quicker. Flame your loop again and sterilize it. And now you have to dry the slide down. You have to dry this liquid off. And you can do it several ways. The easiest way, to me, is just to hold it over the flame, not on top of the flame. Notice I'm able to, the flame is down here, is right here. I'm able to hold this in my hand at this height, and I'm not burning myself. I can feel warmth, but I'm not burning myself. So you have to sit here, and it takes a few minutes, but as you do this, that'll drive off some of the water, and you will have a dry slide. This is the slowest part of the procedure. This just takes a while. I don't know if you can see this too well. Let me see. Maybe against my shirt is the best. You see that little smear there? That's dried down bacteria, and that's all the darker this should be. If it's really opaque and it looks like flaky bits of soap scum or something, you've put too many cells on, and that's going to probably ruin your gram stain. At this point, you've got bacteria on the surface, and you have to heat fix them. Otherwise, when you add the subsequent liquids, they're going to fall off. They're going to wash off. So to do that, it's real simple. You just take the slide and you pass it through the flame. Once or twice, that's it. That's all you need to do. They're now stuck to the surface of the slide. Then you start the actual procedure. At this point, I'm going to turn off the burner because I don't really need it at this point. So the first thing you do is you add copious amounts of crystal violet. You don't have to skimp. You can just put this dropper-wise right onto the surface. And ordinarily, we'll go through this quickly, but you let that sit for about 30 seconds or so. You then wash it off. You can either wash it off with water, or you can wash it off with the next in the series, which is iodine. I'll wash it off with water here just to be a little bit less wasteful here. But all you do is you turn the slide a little bit and just go ahead and rinse it off of excess stain. At this point, everything will be stained purple crystal violet color. Now, because the crystal violet will flow out of a cell relatively easily, you want to make it big and fat. So you add something called a mordant. In this case, it's iodine. What iodine does is complex with the crystal violet and make it so you just put it on huge dropfuls, let it sit there again for 30 seconds ordinarily. That makes a complex with the crystal violet, the iodine, so that the crystal violet will not flow out of the cell easily. Right? So 30 seconds later, you wash this off. And you can either wash it off again with water, which is probably what I would do, just a few drops. Now this step is the next step. It's critical. The cells are still all purple, but now if they're gram negative, ultimately, they're going to end up taking the counter stain, so the crystal violet will be washed out of them. To do that, you use the destain solution, which is basically acetone and alcohol. If it's really a gram positive, this won't wash off wash the crystal violet out. But you have to be careful because you can put too many drops of D-stain and get rid of the crystal violet on a gram-positive and have it look gram-negative. You can also under D-stain and end up getting something that looks gram-positive, but it really was supposed to be gram-negative. So to do this, you put a few drops in here, turn the slide on its end again, and just put drop by drop a little bit of crystal violet on, or D-stain on here, and you'll see in your slide that the crystal violet starts to come off the end. And I stop when the crystal violet comes off basically colorless, and then I immediately, if not sooner, rinse this with water to stop the destaining process. Because the destain is on there, it will keep destaining and you'll get a weird result. Now you're set, because now all you got to do is, if it's a gram-positive cell, they're still purple. If they're gram-negative, it's clear. And if they're gram-negative and clear, you couldn't see them under the microscope, so that's why you add the counter stain. Traditionally, it's saffronin, which is pink-red. Drop that on the slide again, let it sit for another 30 seconds or so. Rinse it off. And then you dry your slide with bibulous paper, basically blotting paper, and then you're ready to go ahead and look at it under the microscope.